I'm gonna welcome everybody to the Environmental Engineering Graduate Seminar. And as usual, if you want continuing education credits for this session, contact Eric Seegren and he can get that for you. Today we are honored to have Dr. Greg LeFevre with us. He's an assistant professor of environmental engineering and science at the Department of Civil and Environmental Engineering, Hydroscience and Engineering at the University of Iowa. He's alumni of our program as well at Michigan Tech. He started at University of Iowa in 2016, did his BS here at Tech, MS and PhD at University of Minnesota, postdoc at Stanford in environmental engineering. The LeFevre lab focuses on elucidating biotransformation products and pathways of emerging organic contaminants with the goal of informing improved design of engineered natural treatment systems for non-point pollutants like urban stormwater and agricultural drainage, and transforms wastes into resources. He has received multiple sources of recognition for his work, including the National Science Foundation Career Award, the University of Iowa Early Career Scholar of the Year Award, the American Chemical Society Editor's Choice Award, and the ACS Best Paper Award, the Royal Society of Chemistry and Environmental Sciences Best Paper, National Academy of Engineering Frontiers of Engineering Fellow, and the AEESP Best Dissertation, among others. And all because you went to Michigan Tech for your bachelor's. That is so awesome, Greg. So with that introduction, Greg, take it away.
Well, thanks so much, Brian, for the invitation to be here and stuff. And it's been really fun to reconnect with. I still think of Michigan Tech as really like home institution and where I got started. I mean, when I started, I didn't totally even know what environmental engineering was. I knew I wanted to do environmental science and I liked chemistry, and engineering sounded like we were solving problems. And so this is how I kind of got into things. And I was really lucky to be able to kind of fall into that environment and particularly get involved in the mentoring environment that is at Michigan Tech, where people are really care about each other. And it's been neat to go wherever I've encountered other tech grads from around the country and stuff like that. There's always a special connection in there, and that is oftentimes what is stronger than any other place that I've been. So it's really fun to be here and there and to talk about some of these things. So oops, I always do that. So with that, I want to first start off by thanking my lab group, because without them, I wouldn't be able to do any of these things. And so I use the royal we a lot, and these are the people that are actually doing the work, right? We all come up with really fun, creative ideas and stuff like that, but they're the ones that are actually executing it. And it's been fun to work with people and kind of working with people and being able to start that kind of paying it forward mentorship process. This is something I'll quote John Gerke about one time. He had done something really, really important for something related to my development. And I was like, I don't know that I would ever be able to pay you back for this. And he said, you never will, you're right, but you can pay it forward. And so this is a way that I hope to be able to do this. And that's the most rewarding part of my job. And so I get to work with really great people. So here's just a few of them. And also our funding sources for the work I'll present today are from the National Science Foundation and the US Geological Survey. All right, today what I'm going to present about is kind of a little bit of an overview of some of the work that we do in our lab. As Brian mentioned to start out with, we focus on understanding the transformation products and processes of emerging organic contaminants in the environment on the premise that if we understand how these processes work, then we can inform low energy natural treatment processes that will be able to help solve some of the problem here. And so kind of the four main thrust areas that our lab works on is, I believe you can see my mouse, is the looking at emerging pesticides in the environment in both agricultural and urban systems, and as well as looking at how complex organic contaminant mixtures, particularly during the water recycling and de facto water reuse, how those change in the environment. And then I also do work with green stormwater infrastructure. What happens to pollutants when they go into things like fire retention cells and rain gardens? And then also along with kind of informing some of the science of the plant systems that go on here, and both a remediation and then also a water reuse and exposure perspective, is we do work with understanding plant uptake and metabolism for water reuse, both from like a phytoremediation perspective, but then also a human health and exposure perspective. So if you have water recycling, particularly like they do more in arid regions, what happens for like to these contaminants when, for example, recycled wastewater is used for irrigation? Do these go in and present a human exposure route? And so today, what I'm going to talk about is really kind of two stories here, and really focusing on some of the work that we've done with pesticides and also complex exposure mixtures. So some of the stuff is actually fresh off the press or still in press. So you can it'll be fun to kind of see some of these things. And so it's not totally polished and everything. So I'll focus on those two areas. And I think they complement each other pretty well. The other thing I put a quadruple asterisk here is because just this past week, I got notification from a USDA program manager that we've just got a 4-year grant funded looking at water reuse and plant uptake for recycled water. So I'd love to have a PhD tech grad to come and work on a project like this. So PhD tech grads are hard to come by because they were recruited by a lot of different programs and stuff like that. So I'd love to have one come and work with me. So talk to me afterwards. Shameless plug, right? All right. So the first part of the talk that I'm going to look at is this work that we've done with pesticides, which I think is really cool. And sort of the central challenge that we've identified with these pesticides is we're trying to understand the fate and transformation of target-specific pesticides from water sources. And we're looking at this from really the source to the tap, kind of that continuum as it goes along from the application to potential for human exposure. And the reason we're interested in so-called target-specific pesticides is that these are being increasingly used because there's what they are is they're really good intentions. The intention is to be able to make pesticides that more closely target just the pest without hurting non-target organisms. This is conferred by what is referred to as the pharmacophore. That's nomenclature from like pharmacology, like that's the pharmacophore of a drug or something. It's the critical functional group on a molecule that causes a biological effect. And so what we are studying is how these target-specific pesticides, if there's a slight chemical transformation along that pathway from the sort of application to source water to finished water, if there's something that can happen to that, does that change its fate, exposure, toxicity, or something? So we've been looking mostly at neonicotinoid insecticides as one example of a target-specific pesticide, but there's others of these as well. So neonics are actually the world's most widely used insecticides at this point in time. And these three that we have here, the three most common ones. This is imidacloprid, clothianine, and thiamethoxam. And they're all characterized by being having really pretty low log KOW values, which means that they're highly water soluble, right? So they'll move a lot from the point of application. And they all have some sort of highly electronegative functional group, either a nitro group like these, or sometimes a cyano group that confers the insect specificity. And so if you actually look at a map of over basically the past 20 years or so like that of the usage in here, these are from stats from USGS, you can see that these really started off as kind of a specialty herb or a pesticide for a few specific fruit applications and stuff. And then they exploded in usage. And we are here in Iowa, which is usually ground zero. This in the Central Valley of California are usually ground zero for pesticides, right? And before I moved to Iowa, I had no idea that corn came in fuchsia, right? And what they are is that most corn in soybeans in the state are planted coated in insecticides and herbicides for the purposes of basically a prophylactic application, so that insects, when they grow, they then that transfers and translocates within the plant and protects it from the insects. Problem is that about 90% of it tends to just wash right off. So it's a cheap way of applying it. It also gets around regulations as far as applying herbicides or pesticides, but it is not very efficient in that capacity. So the kind of one of the take homes that we were looking at here is that we know that alteration of this insecticidal pharmacophore, right, that's nitro group here, changes the insect specificity of these compounds, right? So you can see that kind of in this molecular bonding diagram here, you can see this electronegative group. If that nitro group pops off, it means that it changes the binding specificity with insect acetylcholine receptors, which are these neurotransmitters inside the insect. And it actually makes it so that these bind really highly preferentially to insects and not very much to mammals. These bind so more towards mammals. So these transformation products actually reverse the selectivity of the insecticide. So remember, we have a good intention of a target-specific pesticide, which this is. It's much less toxic to mammals or vertebrates than it is for insects. But upon a relatively small nitro reduction or some sort of alteration like that, it can actually reverse binding specificity towards vertebrates. So not good, right? So just jumping right in here, what we did was we conducted a study in Iowa City. And what we looked at was we just wanted to see what's happening here in terms of is anything being removed of these insecticides, these neonics, during drinking water? The University of Iowa actually has its own drinking water treatment plant from the Iowa River, and which is a heavily agricultural region. And what we found was that neonics, first off, were ubiquitous in the tap water. And if we looked at these, this is a little complicated graph, so I'll explain it. This is the source water. This is the dash is the finished water leaving the plant. And these are the university, and this is the Iowa City. So the University of Iowa versus Iowa City. So first off, you can see that in the university system, there's essentially no change in the treatment system. So basically nothing was removed in a conventional coagulation, flocculation, water treatment system. In the city plant, which has some, it's a same water source, but it goes through riverbank filtration. So the levels initially a little bit lower just from dilution by groundwater, but you still see some, but this actually has a activated carbon bed for taste and odor. And we see substantial removal of these in the city system. So if we look a little bit closer at this conventional drinking water treatment plant, we went along the treatment train and we saw that these kind of the most common three compounds basically persisted throughout the entire plant. It didn't matter where you were along the way. The only real exception to that was that in the following lime soda softening at elevated pH, their thiamethoxam actually did drop quite a bit. And we attribute that actually to hydrolysis. So I'll talk a little bit about that in just a little bit. So this might seem like, okay, well, that's not terribly surprising because conventional drinking water treatment is designed to remove particles and pathogens, right? It's not designed to remove highly soluble emerging contaminants. So in fact, oops, let me go back. The fact that there was removal in the city plant with the activated carbon bed was actually more surprising than the fact that we didn't see any removal in the conventional drinking water plant. So this was, in some ways not super exciting science, but also kind of scared the pants off of people. And it was the first study on this. So this paper got on the cover of ES&T Letters, which I thought was pretty fun. My grad student and I designed the cover for this, and this was really fun. And yeah, here's the drinking water treatment plant, the intake for this. And coming in here, and yeah, you essentially don't see any removal on that. And so yeah, Catherine and I were interviewed by the Washington Post and the BBC, and was the front cover, and we got on the radio and stuff like that. So it was a fun thing, but at the same time, it was kind of like, okay, great, they're in low levels, it's not removed. Is it a risk? We don't totally know that at this point. There's not very good chronic exposure data associated with these. So one of the things that we're doing is following this work. The College of Public Health here went and established a rural health neonicotinoid collaboratory to try to understand these. And some people are looking at things in groundwater, human urine metabolites, and more chronic exposure levels and stuff. So we actually just published a joint literature review that's really long and extensive that kind of follows up and looks at it from all these different angles. So one of the things that we did follow up was that we thought was actually much more interesting science and interesting follow-up was that we know that, remember how we said that these metabolites are actually much more toxic to vertebrates than the parent pesticides are. And we know that these metabolites can be formed from both biological and abiotic processes, like some photolysis, microbial transformation in the environment already. So some of these breakdown products, for example, for imidacloprid, you can lose that nitric group and you get a desnitramidacloprid, and then that can be further transformed down to a urea group. And so we know that these things are formed in the environment. What we were interested in is, okay, well, what happens to these compounds if they undergo, disinfection, right, in the drinking water plant? And so what we did was we did just very simple batch chlorination tests, and then what we did was we used high resolution mass spectrometry to, under these very simple conditions, to do basically a semi-untargeted metabolomics style experiment to try to do product elucidation and MSMS fragment analysis, and for just basic structure elucidation. And so what we did was we found that all these things can chlorinate. The rates are actually different between them. I don't think I showed any kinetics data in here. But what's interesting about this is that the parent compound and the products can actually go in and form the same net result chlorinated products. And so, but when you have the metabolite as the precursor, and then those are chlorinated, that happens much more rapidly and forms all kinds of different things. We know from the literature and all kinds of, that's well established, that whenever you add halogens to things, that's usually not good in terms of increasing the toxicity. But at this point, all we have are knowledge of these things. There's no toxicity data, obviously, about any of them. So remember back about how I talked about that one thiamethoxam kind of disappearing following under that really high pH conditions. So we also followed thiamethoxam as another example. And so what happens with that is kind of interesting, is that the thiamethoxam itself didn't chlorinate at all. It was very recalcitrant to that. But what we found was that under the really elevated pH conditions, it will go in and you'll get a hydroxyl attack on that, and you will form two different hydrolysis products. And one of which, this 248 compound, these are just named after the mass fragments, right? And so this, you can see, okay, here we go, that the thiamethoxam goes away, but we can see that kinetics wise, that the 237 compound over here, the 237, the thiamethoxam hydrolyzes, right? This compound, the 248, is actually recalcitrant to chlorination, and it'll just stay right here and won't do anything. So this is a dead end product, whereas this one actually, you see it forming by hydrolysis, and then it goes away, and then you see the formation of this chlorinated one in there. So I don't have all the kinetics data shown in here, but what's interesting is that when this undergoes hydrolysis and then subsequent chlorination, it actually ends up with the same end product as direct chlorination of the chlorothianidin, which is kind of cool and interesting, but it actually kind of makes sense because in the environment, thiamethoxam can actually undergo transformation processes that go in and form the insecticide chlorothianidin. And that's actually part of the way that the insecticide works, is that it breaks down and forms a more insecticidal compound. So like I said, at this point, we don't know anything about these in terms of whether they're more toxic, how long they stick around, or anything like that. So here are a couple lessons that we learned from trying to understand these kind of novel disinfection byproducts. Well, lesson number one is that these molecules are actually really complex in how they behave, and understanding these in the environment is it's really interesting but difficult, and that makes a difference in how they react. So first off was the role of tautomers and tautomerization in the reactivity of chlorine. So for example, imidacloprid, the parent compound, can be present as both its imine and imine form. And so these are both differentially labile towards chlorination. And the desnitroimidacloprid also can form these two different forms, the amine and the imine. And so these form different chlorinated products depending on the distribution. This is basically comes down to thermodynamics and what is the greater propensity in there. So we can't really necessarily quantify that, and that starts to get into where the position of a double bond is or something like that, and that's stuff like beyond the capability of high-res mass spec and starts getting into NMR type stuff and whatnot, which is I don't do that. I work with my friends in chemistry to play with that. So the second part here, which is probably good news, is that a lot of these products seem to be relatively labile. They don't seem to be probably sticking around that much. For example, if we look at this compound, what we did was we looked at what happened, and then we added a reductant, in this case sulfite, and it back transformed and dechlorinated it. So that could be one reason why we haven't seen these necessarily, or we don't see a rapid formation of these, for example, because any type of reductant that could be potentially possible in the water distribution system or something like that might actually back transform these, and these might actually not be that huge of a risk. And so finally, the kinetics are really important. The difference is that, for example, we know that some of the parent compounds, they did chlorinate, but it was really slow, like half-lives of 70 days, on the order of days, right? Whereas like some of the products were on the orders of minutes. And so the difference between understanding the reactivity of the parent insecticide versus the products is quite a bit different. And that's really important for understanding what the final fate of these is. So remember back to where we looked at how the difference between the university system, which uses a conventional coagulation, flocculation, drinking water treatment, which is what most municipal systems are. But the Iowa City system used to have horrible water with, it smelled like stinky fish. And so what they did was they put in these, you can see these systems over here. Here's Greg, he's one of the plant managers, and they have these activated carbon beds. And they're trying to go in and remove the taste and odor compounds. And so we were interested, we saw in the preliminary data that these are able to also remove a lot of these neonics, which according to conventional hydrophobic partitioning and everything like that, we wouldn't expect that. Remember, some of these have a negative log KOW value, which would suggest that they would much prefer to be in the in the liquid phase and not solving up. So we wanted to break this down because this could be a very feasible removal mechanism. So this is work that is actually not even, it's just been accepted, and so I was happy to add these last slides late last night in here, because it's actually not even on the internet yet, but you will hopefully find this at the end of the week, is that we just had a really cool paper in the S&T that looks at how changes in these neonic pharmacophores actually is a major driver in predicting the difference in sorption propensity, but also the sorption mechanisms for black carbon. Kind of the take-home point here is that what we found was that if you look at kind of this, I mean, this is a kinetics kind of style isotherm here. So we're looking at sorption, and these are the points and then the confidence intervals. We looked at really looking at how the parents and the metabolites differ, right? So this is the one with the nitro group. Remember, highly electronegative, that's the insect binding one. This one has a much more positive charge distribution, and that binds more to the mammalian ones. You can see that the sorption propensity is much less for the metabolite. So that's really important because the more toxic one is not sticking to the activated carbon as much. Not good, right? And so what we want to do is look at some of the mechanisms here. What we found by doing some idealized experiments with GAC and also with these different types of functionalized carbon nanotubes, and I'll look at that, we'll share those a little bit on the next slide, is that we found that the mechanisms were actually different. A lot of what was going on with the parent compounds was actually binding to or having like things like pi-pi binding interactions with the graphene surfaces associated with the GAC, and much more interactions with this end of the molecule. And so those are much stronger. And what we're seeing with the metabolite is that you're getting much more electrostatic interactions with these different types of imperfection charged functional groups that are present on the surface of the GAC. And those are much weaker. And so not only is the propensity changing, but the mechanism is as well. So one of the ways that we really looked and dug into this was we used these idealized systems. So we used, real GAC that was from the drinking water treatment plant. We characterized how that looked and stuff. But we also used these functionalized multi-wall carbon nanotubes. They could be functionalized, or we could have functionalized and non-functionalized ones. And so kind of let's look through here, right? We looked at this parent compound, right? The one with the electronegative functional group. And we looked at kind of how the propensity of these different functionalized and non-functionalized sorbents are. And so the parent compound bound most to the non-functionalized CNTs, which is suggesting that it's binding mostly due to these pi-pi bonding interactions there, because there's no functional groups. And if we looked at what's going on here, the ones that it went the least with were actually the ones that were the carboxylic acid functionalized groups. And that's suggesting maybe there's some repelling action that's going on. And if we look at what's going on here with the more positive desnitroimidacloprid, what's actually happening is we get the greatest amount of sorption onto the carboxylic acid functionalized carbon nanotubes, and the least with the amine ones. So this has positive charge, this is negative charge. And so here we're getting much more charge interactions on there, which tend to be more weak, but that's really important. So we have other supporting evidence as well for this. And we looked at all kinds of different water chemistry conditions and ionic strengths and temperatures and everything too. But one of the big things here is that this helps us understand how this works. And this also helps us understand potentially ways that we could like tune and engineer different types of engineered sorbents that could be useful for these types of metabolites too that are more dangerous. So shifting gears a little bit here is that we know that we also know that these metabolites are forming somewhere in the watershed. We don't necessarily know where. And remember, one of my goals here is to try and understand how we can create sustainable systems that use low energy. So one of the papers that we had last year was we were looking at two different types of neonics, imidacloprid and thiacloprid, which are basically the same difference in their functional groups. And what we looked at was the aquatic plant Lemna duckweed, which is, common all over the place, all over North America. What we found was that it actually facilitated with unsterilized duckweed, so the combination of the duckweed and their associated microbes, we're able to degrade these neonics without production of known toxic byproducts, which is really critical because two things. First off is that a lot of microbial processes go in and generate toxic byproducts, which can be worse than not the parent in any way. And the other thing is that if we looked at just the plant or just the microbes, the insecticides didn't go away. But with the plant and the microbes together, it created a synergistic process that was going and able to break down them. So this is something we don't totally understand yet, but it's a cool way of actually affecting the solution too, which oftentimes we environmental engineers look at problems without necessarily being like, oh, cool, we found something that can solve this, right? So finally, one of the areas that I'm also looking at is kind of different sources of neonic levels in places, right? We've been working really extensively with this partner nearby wastewater treatment plant. And most of the time we think of neonics as being from agricultural sources, which is totally true. That's where the majority of the mass is. But we were actually looking at neonics in this wastewater treatment plant. And it's true that most of the time it's relatively low, but for chronic vertebrate toxicity levels, a lot of the levels that are at these different from the effluent as well as the different downstream locations can exceed both the chronic invertebrate toxicity or the acute invertebrate toxicity levels. And so what we did was we also did some spatiotemporal analysis of these as well and suggested that these different neonics are coming from different places. One of the implications of this is that it's not just agriculture. And because this is it's the fastest growing city in Iowa. And so there's a lot of new infrastructure in here and stuff like that. And so it's separated storm sewers. It's not, like runoff from people's pesticide lawns or something like that. One of the things that we looked at here was that could this actually be from more indoor home usage as opposed to outdoor usage or agriculture or food residuals or something. And that's actually what we're implicating here, is that there is a lot related to that could be from pet applications or from indoor pest applications for controlling ants or something like that, or other things. So this is kind of a different unknown exposure road. So kind of our conclusions here for this first section here is that neonics are present in drinking water. We know that their metabolites especially can form novel chlorinated DPPs. We know that sorption is driven by that pharmacophore, and we see differences between parents and products. And we know that plants and microbial communities can synergistically degrade these neonics, and that wastewater forensics kind of gives us an unanticipated human exposure route. Like, this isn't something people are necessarily expecting because it's actually in their hosts, which is something particularly for people that wouldn't be expected to be exposed, like children in houses. So the second part here that I'm going to talk about is a study that actually just segues right from that previous one, looking at spatial and temporal variability of pharmaceuticals in temperate region effluent-dominated stream. This is really Hui Zhi's thesis work, and she's going to be defending in like a month. So this work is really cool and all done and stuff like that. And it's really collaborative. We've been working, this is funded by a USGS 104G grant, and we're working with toxicologists and biologists from USGS and University of Wisconsin-Milwaukee too. And just as a preview, this is going to be out on the web tomorrow, but this is a cover story for ESMT, so it's really cool. We got to put there and look at our, you can see a little preview of that work that's going on. So really what we were studying here is that wastewater effluent-dominated streams are typically thought of in arid regions where there's not a lot of water is scarce, but the reality is that these are very common and becoming increasingly common in temperate regions as well. So that just means that there's a high amount of wastewater influent, and they're going in and potentially impacting the ecology as well as downstream drinking water plants. In fact, a meta-study published a few years ago found that the majority of de facto water reuse systems were actually in temperate regions. And so this seemed like a really interesting place to study. And so we established this kind of field laboratory study site. And really our motivation here was to try and understand from an ecological exposure perspective. By the time these get into bigger waters or something like that, they tend to be very dilute. And these pharmaceuticals are, they're meant to be consumed by humans. They're not inherently toxic per se, but at low levels, they are known to impact fish, and particularly like on an endocrine disrupting level. So our overall hypothesis was to try and understand how these changing mixture dynamics go along the stream, because we know that there are interactive effects like drug-drug interactions, like synergistic and antagonistic effects. And even though the total concentration may go down as you move down the stream, we don't have a very good idea of how those interactions might be characterized. And so we want to see how that works. So our field site was actually only about 6 miles from campus, which is super handy compared to flying to Alaska or something, right? And we worked with a wastewater treatment plant that actually has a very modern membrane bioreactor facility. We set up 100 meters upstream of there, the effluent, 100 meters downstream, and then at a USGS gauging station that is 5.1 kilometers downstream. And if you look at this gauge, you can see that the flow goes like this. It's this very sinusoidal, and that's reflective of the way that people use wastewater, right? So this stream flow is dominated by wastewater. It's a small stream. We could demonstrate that by, you can see the little pictures, right? It's just a little trickle. And sometimes you'd have, in order to collect your sample, you'd have to dig out of the gravel there in order to be able to collect your upstream. Here, it's a relatively small plant. And then downstream of it, you can see that the plant contributes most of the flow. You can see that here too, we're looking at the flows. And if we look at this flow ratio along the top here, the majority of the time, the effluent comprise more than 90% of the flow, up to 97%. I think the low is like 75 or 57% or something like that. And actually, what's kind of cool from a hydrology standpoint is that if we look at the ratio of the effluent to the DS1 or the first downstream site and the second downstream site, we see that the effluent flow is either at or most of the time above the flows at the downstream sites, which means that this is actually changing a temperate region stream, which tends to be a gaining stream, right? Like most of it's groundwater fed, into a losing stream, and changes the hydraulics of it so that you have the flow is discharging from the stream into the groundwater. So it's acting much more like a desert stream, right? So that's really cool, but it also means that, it means that these contaminants that are present in the wastewater also are moving into the shallow groundwater. This means potential for more hyperetic exchange and everything that way. It also means that this could potentially contaminate groundwater. So this is I don't expect you to read this, but what we found was that we measured over 100 different pharmaceuticals, and we see that they largely persist in the stream. We could go in and look at how the concentrations, total levels are sometimes over 30,000 nanograms per liter. That was our highest level. And you could see that they were very consistent in a lot of ways. The blue is the cool season. The yellow is the warm season. And you can see kind of the flow as it moves along. The more interesting thing, though, was that what we were really looking at was how these mixtures change through space and time. And so this is kind of, I call this the evolving pie graph, right? Is that in warm conditions, this is actually the median mass and the composition. You can see that the median mass in warm conditions is much greater than under cool conditions, but also the composition is most bigger. This big one is antihistamines, right? That's allergy medicine. And I've never had more allergies than and around here, there's just, I mean, there's a lot of pollen, right? And so a lot of people take allergy medicines. You can see that the composition changes even under very short distances, and it attenuates much more rapidly under the warm conditions than under the cool. And so the mixture changes, the composition changes, and the total decreases. We also looked at how from the different sites, so the upstream, the effluent, the first downstream, and the second downstream. And we looked at how the rank order representation, right? There's hardly anything in the upstream, maybe a little backwashing, a little bit of non-point source, but we can see that the representation of these compounds, as we look at the top compounds, changes. For example, venlafaxine goes from the first one to the third one, and citraplan being the fourth or fifth one, that is like completely drops off to almost the last one here, as far as like the top ranked compounds. You can see that this mixture changes due to the differential attenuation along that stream range. And so this is what drives some of the mixture dynamics. So we characterize that the mixture dynamics along the stream. You can see that, for example, for some things like metformin over here, like it's kind of all over the map, right? And for some things like citraplan, it is largely gone by the time it reaches the second downstream station. And so you can see these dynamics change, and that is one of the main drivers for these complex exposure mixtures. So our original hypothesis is that transformation in the stream, so compounds going from the parent to the product, would be a major driver of the mechanism behind these complex mixtures. And the reality is that this was not happening. We found that most of the products that were in the eventual mixture were actually due to just things biodegrading inside the plant and then being released. There was very little in the actual stream water itself, which indicates that in that relatively short distance, it was not transforming. So that was actually a really interesting finding. And so finally, what we did was we connected these with batch tests in the lab by looking at photolysis, biodegradation, sorption, and trying to characterize some of these mechanisms of loss. And so what we were able to show was that we can use batch tests, which are oftentimes actually used to characterize these things for regulatory purposes. And that's good for understanding mechanisms of loss, but it's actually really poor for trying to predict the actual rates in the field. That has really important implications for trying to look at fate, because in the field, we actually saw much greater rates of attenuation than we did in the lab. And so, for example, if you looked at citraplan, that rapidly attenuates. If you look via sorption, if you look at metformin, nothing going on there. And so finally, some ongoing work that we're doing is we are looking at different organism classes and looking at risk quotients, so risk there. And then we're applying those to do stochastic risk modeling and to try and understand what would happen under different flow conditions. So doing Monte Carlo simulations and developing models for that. And then also using these attenuation rates that we apply in the lab, and then going in and trying to compare our models to measured values so that we can look at what's going on as we move along that stream range. So I talk about exposure mixtures a lot, so we're looking at the chemicals and the chemical fate and everything like that, but really we want to know what's happening from an exposure perspective. So the other half of this project is what Rebecca Clapper, who's the director of the Great Lakes Genomics Lab at UW-Milwaukee, her lab is doing. So we sent them samples of water to do lab fish exposures. USGS has done these yeast estrogen assays, and then Blythe. They're also doing RNA sequencing and looking at antibiotic resistance genes and trying to understand what's actually going on, how these exposure mixtures are connecting with fish. So we're connecting exposure with effects. We're also trying to do some cool stuff we just got a brand new high-res mass spec and Orbitrap. So we're trying to take some of these big data from the RNA sequencing and match it with our big data from mass spec work. So last year, and this is kind of the end here, but really cool pictures, is that we did a cage fish deployment. So that's Emma, a student from Milwaukee, and we put all these fish in cages here. And then we did an intensive 96-hour experiment where we took all these lab-reared fathead minnows and then exposed them in the stream and measured the chemicals and all these things along the way at the same time. Then what we did was we kind of did this crazy intense working where everybody did all hands on deck to try and collect the fish, and we swabbed them for looking at antibiotic resistance genes, and then dissected them before and after and along the way. And we collected their blood and their brains and fish and liver, and I was just a pair of hands at that point in time because I had no idea what I was doing. I was cutting off fins. So that work is still going on, but it's pretty cool. And we also did some fun fish stocking work with the USGS, and we were really surprised. This is an effluent-dominated stream with 30,000 nanograms per liter of pharmaceuticals. But what's interesting is that we also found that the wastewater, didn't have, it had imidacloprid, but it didn't have atrazine, which the upstream concentration did. And it actually had some really good, this is it's a state endangered, this is called the Johnny Darter, a special minnow that was there. So we found some of these cool things. So our conclusions here really is that mixtures varied along the stream reach, and this created unique exposure conditions where these types of drug-drug interactions that are powered by the mixture are really important for determining ecological exposure. So finally, I'll close with these take-home points, is that that slight structural changes to emerging contaminants in environmental systems and engineered systems they can change their toxicity, the fate of the compound in the environment, the reactivity during treatment, and the sorption propensities. And these all can be looked at from a fundamental basis, but are sometimes actually really difficult to predict. And so we're also looking at how these mixtures come together, and how they change in space and time, and how we can model that to better assess risk for ecological systems, as well as potential for both de facto and purposeful water reuse. And so I'll close with my shameless recruiting plug that I would love to have a Michigan Tech grad, to somebody that's finishing a master's or a bachelor's or something, to come work on a PhD in my lab, because tech grads are the best. So I'll close with that and leave the slide up so that you can play with, or if you want to have questions on this too.
Yeah, thanks so much, Greg.
Hopefully
just imagine a thunderous applause for you for
Yes.
this. Over Zoom, it's hard to see the clapping, but
there we go.
I really do appreciate it. And if you were here, we would be standing and cheering you on. But any questions from anybody for Greg?
Hi, Greg. This is Noel Urban. Really enjoyed the talk. Lots of exciting work there. I had a question about your hazard quotients and what you were doing with those. So were you calculating cumulative hazard quotients and seeing how those varied, or?
Yeah, so this is a really simplistic way of looking at this. And so what we wanted to do with that was to go in, and we took established literature data as far as effect levels in terms of like LC50, EC50 type of things as far as that. And then we just took our measured data and we pulled everything from the entire 5 kilometer reach. And we use that as the inputs for our Monte Carlo model. And that worked really well for the compounds that tended to persist throughout the whole reach, but it neglects the spatial differences that happen for some of the more both sorptive or labile compounds, because what it does is that it overall, it just gives you a more spread distribution for your model, as opposed to, and it underestimates the risk closer to the effluent outflow, and it underestimates it closer, and it overestimates it farther along because there's a much more rapid attenuation. The reality is that in order to be able to use this for modeling purpose, and this was kind of a more proof of concept type of thing at this point, was that you would have to have I mean, we have a decent amount of data at each site, but we don't have flows data at each site. We have a lot of data where the gauge was, and then when we sampled, for some of the time, we were able to go in and get flows data, but that required having a USGS technician be able to come out and collect those and stuff like that. So that's kind of where those were. So the model is oversimplified for that, and it works great for some compounds, which actually was the majority of the ones for kind of more detailed analysis. But for some of them, like the citraplan, it doesn't work.
Great, thanks.
Yeah. And it's weird calling you not Dr. Urban.
Any other questions for Greg?
I have a question, Greg. This is Judith Perlinger. I enjoyed your talk. Thanks. Back on slide 15,
Sure.
where you're talking about the pharmacophore sorption.
Let me see if I can. I don't know if I can go back to. Yeah, well, why don't you explain the question while I try and get back?
So I'm just thinking about sorption by pi-pi bonds, like you were talking about.
Mm-hmm.
And it seems like for that example, at least, there's the core that you're saying is sorbing, so the phenyl ring on the left, and then there's this part that sticks out on the right. And my question is, doesn't that hinder sorption? It just is a bulky group that prevents the pi systems from aligning.
Yeah, and I would agree. Yeah, that a metazole ring on to the side is, yeah. I mean, it definitely would hinder it, and it's not, a perfect benzene ring or anything either. And this is kind of our working hypothesis for what this is. I mean, it seems reasonable based on some of the literature context and some of our some of these equations that or some of the experiments that we've done with these different functional groups. And one of the things that we tried to do also, and I didn't show this, but we also looked at in the Iowa City plant that has the GAC. They were changing out a filter, and they gave us different cores of the GAC. And so what we were able to do was look at desorption of these. What we did was we used a whole spectrum of different polarity solvents to attempt to desorb off the different compounds off of them. And basically, I mean, from a human health perspective, they don't desorb very well at all off in just water. But based on, we did some kind of like PPLFER model type things and thermodynamic looking into in terms of trying to predict what solvents would be optimal for desorbing these types of compounds. And based on some of the things like the Abraham parameters and stuff like that, it seems as though these types of medium, like apolar protic compounds that are would be best at desorbing them as far as like prediction. And that's also what we observed too. So it seems, I mean, based on those, for example, yeah, like you said, the Abraham parameters, it appears as though some things like the ability for example, the ability to induce a dipole moment in the molecule is important, highly correlated in that model. And so it seems like this is kind of our working hypothesis, but I mean, we don't obviously totally know about that.
Okay, so, great, thank you.
Any further questions? I heard something, Greg, and that is I heard antidepressants can get in the environment and make some species very happy, and they're not afraid of their predators anymore, and their numbers are dwindling because of that. Have you heard of that mechanism? Or?
I have heard of that. I don't. I usually let the toxicologist answer those questions, but
Yeah.
I do know that they usually look at or quantify those, and it's been interesting working with the eco-tox people, and I love working with them because they are the ones that usually answer the so what questions. Sometimes I joke about because this is I joke that this is my favorite project because I went into environmental engineering because I want to help fish, not people, right? Everybody's talking about human health and all that kind of stuff, right? But the but usually they quantify it. There's very quantifiable ways that ecotox people look at. They call it predator avoidance behavior. And so they look at like, what is the reaction time of a fish under different conditions and stuff. And yeah, it's really, I think, difficult for them to tease out chronic exposure levels and stuff like that. And that's what these things are, is that, when we did the simulation modeling, even when we include what we call the all flows conditions, it decreased some of the risk, but it didn't decrease it below levels of concern. And so just more dilution from occasional runoff events wasn't significant in this effluent-dominated system. And so it's true that by the time this gets into, for example, the Iowa River and it's diluted by 150 times or something, the levels are probably not that high, but they're, as far as for fish or for people, like the downstream drinking water intake, but certainly for a localized stream system, these do make a pretty big difference. And some of these things are, pharmaceuticals get introduced into the market all the time. And so we've actually the ecotox collaborators are really interested in this one antihistamine phenylhexidine that we've been looking at pretty extensively, as well as metformin, which is the highest compound. It's an anti-diabetes one that is, it's the highest compound concentration that we saw all the time. And so these things have kind of very low level chronic effects that are not really well characterized, but certainly will have a difference. And we know for sure that characterizing these complex mixtures is both difficult and pretty uncertain, and especially looking at the drivers of these.
Yeah, very complicated. Any last questions before we go?
Well, just one other, Greg. It's really surprising to see such enormous flow modification by the discharge. You would think that that would be against the permit requirements. What's your sense of how frequent such large discharges are into low flow streams?
So, obviously, my knowledge on this is based on just knowing the literature on this, but I was surprised by that and how frequent this is. This is something, the effluent-dominated streams, everybody thinks of them as being an arid system phenomenon. And, in terms of both their presence and their impacts and stuff, but this is a pretty extreme example because, like I said, most of the time it was 90% plus treating effluent. But this is actually really reasonably pretty common for a medium, size municipality. Yes, I said it's the fastest growing city, but it's still not a huge. There's only like 25,000 or 30,000 people or something like that in North Liberty, and the next town over has its own treatment plant, which goes into a slightly larger one, and Iowa Cities, which is 100,000 people, goes into the big Iowa River and stuff. But in some of these small ones, for the same reason why, they're not getting their drinking water from the main river, they're getting it from the ground, and they're discharging to a small stream. It's actually really pretty common. And so they're having something where it's 90% plus, but having something where it's 50% plus is really common. And so, yeah, some of the work that the modeling work from Jacqueline Rice and Paul Westerhoff has been really illuminating on that. But I also have to say that some of the things here, we're balancing some of the, the positive and negative effects here. It's like, yes, we're interested in the pharmaceutical effects. There's been work out of Europe, particularly in, I think, in Switzerland, Germany, but their position was that pharmaceuticals are important, but that most toxicity, particularly for invertebrates, is driven by pesticides, which is true and important. And in fact, in these type of things, we use atrazine as kind of a background thing to look at the upstream agricultural influence, which very low flow, but you would get some influence from that. And for example, the wastewater effluent diluted the atrazine so that it was very, very low after that, which was beneficial. I mean, it contributed some pesticides, but it diluted other ones. This is a really state-of-the-art membrane bioreactor. It had, we did a few tests for like E. coli, and there was nothing in there, so it diluted out the E. coli, and it does biological nitrogen and phosphorus removal and everything like that. That it was actually in a lot of ways, it modulates the temperature, and the water that it puts in is actually cleaner than the water that it displaces for some things. But for others, it definitely does not. So, but the reality is that this is way exceeding its permit requirements in terms of when they built the plant. They kind of did this really cool demonstration project and stuff, and and so it's doing really well. And most plants that discharge these small systems are not performing at this high of a level in terms of all these other constituents that they're doing. But even at a really high performing plant, you still are getting tens of thousands of nanograms per liter of pharmaceuticals that are putting in, because again, they're not designed for that.
Yeah, thanks.
Okay, one last question. We ran over, but the discussion is really good. So any last question, anybody?
I have one quick question. Hi, Greg. Thanks for the talk. It was very fun. You mentioned earlier how because of the downstream flow is so much higher, the water's going into the groundwater. Did you look at or did you see any evidence that these pesticides are accumulating there in the kind of the vadose or hyperureic areas?
So the short answer is we did not for this study. There's a brand new hydrogeologist that got hired in the geology program, and I'm trying to see if I can wrangle her into doing some work with us and stuff. But the longer answer is that the USGS collaborator I have on this project is Dana Colpin, and he has done a lot of work in these types of systems with emerging contaminants, and they worked at another system kind of similar. We didn't, not as extensive of a study or anything, but they did do some like little piezometers in some of the places where you would expect it in hyperureic zone and shallow groundwater exchange. And yeah, even in like tens of meters back, you could look at some of the things that are particularly, like, for example, the anticonvulsant carbamazepine is really recalcitrant, but also poorly sorptive. And so it will go through the shallow groundwater and go into there. So I don't suspect that it would be a major like contaminant type of thing or something like that, but it is pretty interesting that a lot of the flow is recharging into the groundwater too. So it does, I mean, effluent-dominated streams I think are a really fascinating thing, because in temperate regions, it not only changes, you not only have this dilution effect and you have this inputs of these all these different pharmaceuticals, but you also are fundamentally altering the hydrology and the stream hydraulics such that you are changing gradients, and you have it's acting much more like an arid losing stream. I mean, it is a losing stream now. So I think it's really fascinating.
Agreed. Very cool. Thank you.
Yeah, to be able to connect the exposure with the effects is really fun. So
yeah, well, very good discussion, everybody. We ran over, but it's been really good, and thanks again, Greg.
Well-
Fascinating talk, maybe lead to some collaborations, who knows? But we really appreciate your time.
Well, thank you so much for the invitation, for having me, and please feel free to follow up with anything too.
Yeah, it'd be great. All right, everybody. We'll talk to you later. I'm going to end it now.